All right, good morning. Can you hear me? Perfect, thank you. All right. Before we start, does anybody have any questions before we begin? This is like my Monday, I suppose. Yes.
We were expected to have a quiz soon over mainly chapter 2 material and chapter 3 material.
It's only going, yeah, it's going to be mainly chapter 3 only. And I'll tell you when it is. It's not quite yet, but it'll probably be next week. Monday or Wednesday next week. All right. Anything else? There's an issue with the first lab. I was also sick on Monday, so that was helpful. But it turns out I don't have some of the reagents for the lab. And the lab is a linked lab to the next one. So it's not like we can jump, right? It's 4 weeks in total. I've ordered some reagents that I hope will then supplement what I already have so that I can run the lab. So if the kit doesn't come in this week, it's not the end of the world. I can still do it next week. But I'll talk to Richard about how we can set it up in Bainbridge, because Richard will have to get some of the resources from me that I can supply this week once I get things rolling again. Any other questions? You should be starting to work on your presentations, right? You should be looking into and the material on the particular technique that you've been assigned, start putting together slides and figures that you want to show, right? Aim for between about 10 and 15 slides. Don't go any more than that because you run out of time. And even at 15 minutes, you know, so 15 slides gives you a minute a slide. Important to practice, right? Just in your head or out loud to somebody else. Make sure you're on time, because when you'll find when you speak, you tend to waffle and you'll go off target, and before you know it, you're halfway through and 15 minutes is up, right? Or you go too fast and you explain the whole thing in 5 minutes and nobody gets it, right? So it's a balance. All right, so I'm going to continue where I left off. I don't think there's any questions from Bainbridge, so it's good. And it's chapter 3, and I was talking about, why isn't this working? I left when I was talking about predicting tertiary structure of proteins using computer algorithms, which has been very, very successful. I'm going to continue now talking more about tertiary structure of protein. Fundamentals to remember, yes, is that primary is the sequence of the amino acids, secondary structure is discrete folding of elements within that structure, like alpha helices and beta pleated sheets. And then a tertiary structure is association of those motifs together that form the native protein. Native protein is stabilized by interactions, right? Remember those interactions. You've gone through them many, many times before. Things like hydrogen bonding, ionic bridges between charged amino acids, and hydrophobic interactions, as well as van der Waals forces of attraction. You'll see as we progress through the course that protein structures can be represented in many different kinds of models, right? From simple ones where we're just looking at the shape of the peptide backbone. Okay, alpha helices are noted. I don't know why it's so, some of my pictures are not clear, but they are clear on the slide. So I'm sorry, I think it's something wrong in the presentation. Maybe it's because it's in Teams. This is a much more complex formulation of the model, but it shows the R chains together. So that's why it looks so much more jumbled, because we see in the R groups as well. The most common form that you'll see represented in the book is the ribbon diagram. And the ribbon diagram is useful because it shows the direction of the chains. And that's helpful for anti-parallel and parallel beta sheets to look at their conformation. We then have a surface area model, right, which looks at the local hydrophobic and hydrophilic kind of topology of the whole protein. And we can learn from this type of view potential interaction domains on the surface of the protein that are likely to then co-assemble with other proteins through that interface. And then this is a hybrid model which contains both the kind of water accessible and the ribbon diagram together. And so I'll be referring to these kinds of models repeatedly as we go through the unit. So primary structure, the amino acids, leads to local secondary structure elements forming. And those secondary structure elements are things like alpha helices and beta pleated sheets. And then how those secondary elements interact with each other forms the tertiary structure. Now, within this tertiary structure, I want to introduce you to an important word, which is domains. Domains of a protein are secondary structure elements that form and coalesce and are responsible for a certain specific function of the protein. You learned in genetics and in biochemistry that proteins are composed of multiple domains that lead to the function of the protein. Things like, for instance, a transcription factor. Right? A transcription factor has a domain that has to be able to recognize DNA sequences and bind to them specifically. And then it also might have a domain that allows for the protein then to couple with another protein. Okay? These domains are often trying to think of a better word, separatable, that's not right. Discrete. Like discrete secondary structures. And you can often dissociate them from the whole protein. Like, for instance, transcription factors, we can manipulate them in vivo and only express certain parts of the protein, like the DNA interacting domain of the transcription factor. And what's amazing about the domains is that they retain that function even in the absence of the rest of the protein, so that these domains fold up and are responsible for the function and separatable from the rest of the protein. Most proteins in eukaryotes have multiple domains within them that can either be structural in nature, so requiring the association between proteins or between protein and DNA. They can be functional domains, like for an enzyme that has catalytic activity, that domain can be isolated from the rest of the protein. And it's very useful for us as molecular biologists because we can look then for the presence of these domains in other proteins. Right? We've sequenced approximately 250 million proteins, right? But the process of then determining what the structure is for that protein is very, very complicated. Okay? X-ray crystallography or X-ray diffraction studies is the norm for looking at large protein structure. But trying to get large protein structures to crystallize to allow you to determine their structure is very, very difficult. They only crystallize under certain conditions, and these have to be manipulated in the test tube. And so there's a lot of trial and error involved. So it's not a simple process. It is a simple process to put that sequence into an AI algorithm and ask it to then generate a likely tertiary structure for the protein. What is it doing when it does that? When it does that, it's looking at the domains of proteins that have already been crystallized and the structure has been determined. And so we look for similarities between the domains that allow us then to determine what the likely structure is of it. Okay. And the example that's given in the textbook to highlight the domain structure of proteins is the influenza HA protein. It's called the hemagglutinin protein subunit of the viral envelope, right? And so we don't see the whole protein here, but if you imagine the protein, the virus being encapsulated by a membrane, okay, here. And then these proteins are then studied on the surface of them. Remember COVID, they called them spikes on the surface of the protein, the virion, that is responsible for binding to receptors on target cells. Right? This is a single polypeptide chain that has two domains to it. It has a structural domain here, and it also has a globular domain right here. The structural domain is responsible for giving it structure and function, right? With the globular domain is the site of interaction with, the target. The protein is composed of three of these polypeptide chains then that assemble together to form the native protein. And we see this again and again. Another example of modality of domains in proteins is the following example. Cells have to be told to divide. Yes, they're given specific signals by growth factors that the cell responds to divide and actively enter into the cell cycle and proliferate. On the surface of cells, they have these growth factors, and they're tethered like this. It forms one polypeptide with separatable domains. And the separatable domains are called EGF factors or epithelial growth factors. Okay? And the way that the signal is released is that a peptidase here cuts the individual EGF proteins from this long chain, and it snips them off, and then the EGFs migrate and activate cell division. And we see this domain in other proteins as well. This protein is a cell surface receptor that contains the EGF domain. This is plasminogen activating factor, which is responsible for blood clotting. It also has an EGF domain within the protein. Because of their evolutionary relationship to each other, it's very, very helpful for us to compare the sequences of proteins to each other in order to look at their similarities to other proteins. Yes. So in this example, we're looking at the similarity between proteins that form hemoglobin and myoglobin, which are evolutionarily related. Right? Myoglobin probably underwent a gene duplication event resulting in two genes, alpha and beta, that give rise to the two polypeptide chains, alpha and beta, that make up the heteromeric tetramer, which is hemoglobin. And that the plant hemoglobins are also quite similar to the animal hemoglobins and probably originated from an ancestral myoglobin protein, millions of years ago. Similarity with proteins means that we categorize them into families. And those families exist based on similarity between amino acid sequence and structure and function. On the right here is a map of about 500 kinases that are found in the human genome. It's about 480 human kinases. What do kinases do? Kinases add a phosphate. Right? So ATP, ADP, and the phosphate gets added onto the protein. The similarity between the proteins means that they fall into families and subfamilies, which shows relationships to each other. This one is the tyrosine kinase-like proteins, which specifically phosphorylate proteins on the tyrosine amino acid. No, sorry, tyrosine kinase. This one is tyrosine kinase-like. I'm sorry. So sequence-wise, they're structurally similar to the tyrosine kinases, but represent another subfamily, right? Together, these two families make a superfamily. And so all of these derivations just mean relationships between the proteins based upon their sequence similarity. Based on their structure, proteins fall into several groups, right? Globular proteins. What's another one? What's another example of a category of protein? There are only four. Globular proteins. Fibrous proteins. Globular proteins are examples like enzymes. Right? Name describes what they look like. They look like a molten blob, usually spherical or some kind of shape derivation of spherical, right? Fibrous proteins are things like collagen. Pool is running out. Anybody think of another one? Integral membrane proteins or transmembrane proteins. And most so things like cell surface receptors, channel proteins are integral membrane proteins. And then finally, hardest one, disordered proteins. Disordered proteins represent a subset of these proteins because disordered proteins are not easily predictable based upon their amino acid sequence. You cannot accurately predict the tertiary structure of a protein that is disordered. And an enormous amount of proteins contains disordered domains. I think it's about 70% in eukaryotes. And disordered, meaning the secondary and tertiary structure can't be predicted, right? And they often act as binding sites. Let's see. Go from disorder. They contain binding sites for other proteins. So say, for example, helix, and then a bunch of beta sheets, anti-parallel in nature, right? Forms a domain. We often see disordered regions as linker regions between separatable domain structures. Okay, so that this disordered part is not part of the native structure of the protein, right? And this is why we can often separate the domains of proteins, right? Because if you've got this intervening region, just get rid of that, that domain still has wild type function in the sense that the domain's function, say, is binding DNA, it still binds DNA. Right? But contains a region of disordered structure to another domain structure, another domain of the protein. The example that's given on the board is an association between two proteins called Puma and MCL1. The binding of this protein to this protein induces apoptosis and cell death. Okay? But the Puma protein is an intrinsically disordered protein that doesn't form a secondary and tertiary structure on its own. It only forms when it binds and associates with its target. And when associated with the target, forms a structured domain. Right? So the formation is dependent upon the interaction. Right? That's why it's very, very hard to predict. Okay? Because you have to see it in motion, you have to see it happening to show that the two of them can bind to each other. So even though we've come very, very far in determining protein's tertiary structure, we're not there yet. Okay? And we often have to supplement with interaction data as well to help predict tertiary structure. Another example of binding sites are things like histone tails. Why are histone tails so important to the conformation of DNA? Remember that DNA binds histone proteins. Histone proteins form the nucleosome core around which the DNA is wrapped. Remember that? Yes. Cylinder, often drawn to represent the nucleosomes, and the nucleosomes composed of H1A. No, it's not. Reset, get back to my genetics. H2A, H2B, H3, and H4. And that those histones form nucleosome around which the DNA is wrapped, yes? The histone tails are disordered regions of the histones. They don't form a tertiary structure. They hang outside of the protein. and that those histone tails are highly modified. These modifications allow proteins now to bind to and interact with the nucleosome DNA structures, right? They contain docking sites for proteins. Again, binding. Many proteins form supramolecular structures within the cell, right? We went over transcription in depth in genetics, and the pre-initiation complex, which drives transcription initiation, is composed of not only RNA polymerase, but a whole host of general transcription factors that help the DNA polymerase to start the transcription process. Because there are so many different transcription factors that work together, they form these supramolecular structures. And continuing on from that idea of supramolecular structures, I want to introduce you to a new concept which probably haven't come across before, which is the idea of biomolecular condensates. Now, in the eukaryotic cell, right, the beauty of the arrangement of the inside of the cell is that everything is compartmentalized for the most part, right? The ER surrounded by a membrane, the Golgi apparatus, the mitochondria, and so forth, so that we have these compartments that are responsible for certain functions. Prokaryotes do not have such compartmentalization, but they do form these supramolecular condensates, which are like subcellular organelles, but they don't have a membrane around them, right? They are membraneless. And that's what makes them beautiful, is that they can form through associations between proteins and even RNAs and also DNA. And really, it comes down to a high concentration of these units that come together, right? And it's called a liquid-liquid condensate. And depending on the concentration of these molecules, can be either kind of in a liquid phase or a semi-liquid, or even migrate into a semi-solid within the cell. And there are examples in female oocytes in which such a body forms. It's called the Barbiani body. And we see it present in all oocytes, from insects up to animals. Another example in a nucleus, right, is the nucleolus. That is a biomolecular condensate. It's membraneless, but because of the concentration, it separates out from the rest of the nucleoplasm. Okay? The nucleolus is responsible for doing what? Anybody remember? The nucleolus is the site of ribosome biogenesis, right? Makes ribosomal RNAs, and then the ribosomal RNAs assemble with the protein subunits to form the ribosome. This means there's a highly condensed gelatinous mixture of DNA, protein, and they all bind to each other, and I'll show you how. Examples of condensates specifically, things like the centrosome. Centrosome is the microtubule organizing center of the cell, and they're composed of two centrioles, but they're surrounded by like a periphery of proteins, and it behaves much more like a membranous organelle. Nucleolus, like we just described, formation of ribosomal RNAs. Also tRNAs and SNOs as well. Don't realize. And then finally, formations of stress granules within the cell. When the cell undergoes stress due to things like pH changes or temperature changes, then within minutes, these condensates form within the cell that arrest translation and stop it until the cell reaches homeostasis again, in which the stress granules then dissipate. The beautiful thing about these condensates is that it allows the movement in and out of proteins. So proteins can join and then proteins can be released. I don't expect you to remember, specific examples that are on the board or in the book. Like, I'm not going to ever ask you, what does Puma bind to in order to initiate apoptosis, and why is it so special? It's a very open-ended question, and you'd have to remember this, and that's not my kind of style. I would give you the information that you needed, and then the concept would then hopefully lead you to the answer. Here you can see the formation of these liquid-liquid phase condensates, right? Normally, under situations of non-stress or when the signal is absent, then these molecules are dispersed throughout the cytoplasm, but something happens and it initiates a response that results in the condensation. In this example, we've got the visualization of two different proteins. One of them's called APC2 and the other one's called axin. Both of these proteins are highly involved in a developmental pathway that is regulated by Wnt. And developmental pathways regulated by Wnt have been shown to be important in insects to humans. Right? The condensates form through proteins that oligomerize with each other. All right. Oligomerization. A good example of oligomerization are actin filaments. Right? Actin filaments are microfilaments. Yes. They're part of the cytoskeleton. And they form by individual actin proteins binding to each other and forming long strands or chains. In the case of actin myofilaments, it's composed of two strands that kind of wrap around each other. Trying to do that, but failing. They wrap around each other. The process, oligomerization, is driven by domains within the protein that allow that oligomerization to happen. In addition, extensively disordered domains. So in this protein here, APC2, there's more than half of the protein is disordered and doesn't form a tertiary structure. But within that polypeptide chain, there are a number of sequence motifs in here that are able to interact and bind with other proteins. So they're almost like docking structures within the condensate that allow for interactions. And in this example, we've manipulated these two proteins so that they contain different fluorescent molecules. One of them contains a fluorescent molecule called GFP, which stands for green fluorescent protein. And there's another one. It's a tweaked version of the green fluorescent protein that changes its changes its fluorescence from green to red. And it's called red fluorescent protein, right? And we label the two proteins with these two different colors because we can see the colors using a specialized microscope called a confocal microscope. Confocal microscope then gives you the positions of the green fluorescent protein and the red fluorescent protein. If you superimpose these on top of each other, you would see that they coalesce, right? And that everywhere where you have green, you would have red, and usually you would represent them as a merged image to show that all the speckles are orange, where they contain both red and green. The condensates are very, very dynamic. They can form very, very quickly, and they can also dissipate just as quickly. Right? This association requires a phosphorylation event by a kinase to initiate the condensation. And the kinase is activated by a hormone. So in this instance, we give the hormone to the cells, all right? And so this is at time 0, where you can see the GFP here is diffused throughout the cytoplasm. It just looks like a fuzzy green. And then 100 seconds later, after the stimulus, you can see then the formation of the condensates. Next, we're going to describe some structural changes that can occur within proteins due to isomerization of the peptide backbone, right? So what we have on the board is the peptide bond, right? The peptide bond, as you know, has double bond characteristics, right? That don't allow rotation around the carbonyl and the amide, yeah, locked in position. That means that we have the potential to form now geometric isomers. And if you remember, geometric isomers are related to each other through geometry and represented either as the cis or the trans conformation. For the polypeptide backbone, what this means is, what are the positions of the peptide coming in and the peptide going out? Right, from this peptide bond. And so if they're on opposite sides, then they're called trans. If they're on the same sides, then they're called cis. Okay? If you look at proteins, 99.7% of the peptide bond is in the trans configuration. Oh, yeah? But a small subset of them, like 17% or so, are in the cis configuration. Because of the absence of rotation around the peptide bond, it means that the whole peptide is limited in its rotation, or the conformations it can form, right? Based upon only the bond between the alpha carbon and the amide, and the alpha carbon and the carbonyl, right? And these form two planes that can rotate, and the rotation is given by two angles that are called phi and psi. Yes. In reality, the number of different combinations that a peptide can form is completely dependent on the R residues. Right? Some of those R residues are really rather large. I think of tryptophan, cyclic. They take up a lot of space, which means that the conformations are limited. You go through Ramachandran and his plots, in biochemistry. And showing you the topology that's capable for each of the individual amino acids. Back to this idea now, that interestingly, in the 17% or so that you find the peptide bond in the cis configuration is because at this Y position, there's a proline here, right? And a proline has special characteristics because the proline molecule is covalently attached to the peptide backbone. Yes, it links back to the carbonyl. In the eukaryotic genome, there's about 50 or so enzymes that are able to catalyze the conversion between the trans and the cis configuration of the peptide backbone. Okay? And these proline hydroxylases, or proline isomerases, I'm sorry, can dramatically change the position of certain elements within the protein. So in this example, if that proline at that position is in the trans configuration, the loop domain here is in a completely different conformation than when it's in the cis form. Cis for the peptide backbone. And there are many examples of transcription factors that are turned on by this event. Yes, all about switching. How do we get a protein? How can we regulate it? There's lots of ways that we can do it, and isomerization is one of them. Yes, another example would be phosphorylation. Right? Phosphorylation of proteins often leads to their activation, and again, it's through a rearrangement in their structure. The addition of that phosphate causes a structural change in the protein. Alfredson famously showed that proteins are able to form tertiary structures independent of any other help. Yes, and he used an RNase enzyme to show that. And what he'd basically shown is that the structure of the protein is an integral part of the sequence of the amino acids. Right? Now, in reality, this isn't in a test tube. Alfredson did it in a test tube where he was just looking at, one protein in isolation and looking at how it denatures and how it's able to renature and form the native structure, right? But in the cell, there's millions and millions of other proteins, and they're all different. Not all different, but there are many, many different proteins. So it's a different scenario looking at protein folding in that respect. And it turns out that about at least 80 or 90% of proteins in eukaryotes require help in order to be able to fold, right? So the folded state of the protein is helped by molecules that are called chaperones. As you will learn shortly, the chaperones are split up into two categories. One of them has got the same name, chaperones, and the other one's called chaperonins. Basically, chaperones help with folding of proteins, and they do it in an ATP-dependent manner. They use the energy of ATP in order to help the protein attain its nascent folded shape. Some examples of chaperones are things like HSP70. Okay. Where the HSP here stands for heat shock protein. And heat shock proteins are ubiquitous in biological life. They are present in bacteria, and they are present in all other cells of eukaryotes. So they are central and important for not only achieving the folded state, but also retaining the folded state. When a protein folds, it's like a jiggly blob of jello, right? It is easy for it to unfold as well if the conditions change. And so these proteins were initially discovered under heat shock conditions in bacteria. Therefore, they were called heat shock proteins. And they respond when the environment changes. So when the temperature changes in the environment of the bacteria, it goes up. It can cause proteins to now unfold if they just change by a couple of degrees, the temperature. The cell then expresses many more heat shock proteins in order to help retain the conformation of essential proteins required for cellular function. The HSP protein has homologs. Remember what homologs are? Proteins with extensive sequence, extensive same sequences present within proteins that indicate they have a common ancestor or resulted from gene duplication events. And those homologs, they're present in mitochondria. Which is, I can't remember what that, maybe it's HSP40. Not sure. And then in the ER, there is a homolog that's called BIP or BIP. So, so chaperones maintain the folded state and also help to resolve aggregates. Aggregation of proteins usually occurs due to the hydrophobic interaction, right? Some parts of the proteins are extensively hydrophobic, hate interacting with water, and like to shield themselves from the aqueous environment, yes? But what can happen is if another protein also has a hydrophobic domain, these proteins can stick together like so. Now imagine thousands or millions of them, and they can form large plaques, which can lead to disease, things like Parkinson's and Alzheimer's disease. All right, I'm going to stop there. Sorry, I went over a little bit. The next couple of lectures are going to be mechanistic heavy, so please read the chaperones, their predicted molecular function, and also I'm going to be going over an example of an enzymatic reaction, that is the trypsin protease. And I'll see you on Friday. Hello.
Make sure.